Welcome to the fourth lecture, Chapter 4, Tissues, the Living Fabric. This last lecture rounds out the information required for Lecture Test 1. Lecture Test 1 obviously covering the first 4 lectures, and hopefully this one is a little bit more review because this is what has been the focus of the first lab test as well. So you've been covering some of this already through watching some of the lab PowerPoints or looking at some of the lab PowerPoints. But I'll go into detail as much as possible and help point out some things that are going to help you with both lecture and lab tests. So a tissue is defined as a group of cells that are similar in structure and perform a common or related function. And our 4 primary tissue types interweave to form our body. And the 4 primary tissue types are epithelial, connective, muscle, and nervous. And we're going to go through each of those 4. And next to each of the 4, you have a very basic definition of what their general role might be. So we started back in Chapter 1, talked about tissues being organized into organs, and really most of the organs contain all 4 tissue types, and how the tissues are arranged kind of determines the structure and the capability of that organ. And the study of these tissues is known as histology, and it complements the study of gross anatomy, and really provides us with a basis for understanding kind of organ physiology, because how these tissues interact is going to be what allows the organs to interact or do their specific job. So we start with our epithelial tissues, and these are our basic covering type tissues. It could be lining, it could be covering, they could be glandular tissue. With the glandular, it's going to form glands. With our covering and lining, we're talking about our free body surfaces, and our basic functions are to protect, absorb, filter, or secrete, or a combination therein. I'm going to go over some more of the specialized characteristics of epithelial tissue in the next slide. So we have a number of special characteristics for epithelial tissues. They're going to fit closely together to form like a continuous sheet, and it could be 1 to several layers of closely packed cells in thickness, forming that continuous sheet. They always are going to have one free surface exposed to a cavity, a lumen, or a surface known as an apical surface. And then the lower surface is going to rest on what's known as the basal surface or the basement membrane, which is structureless material secreted by cells and really kind of attaches to the underlying connective tissue. Epithelial may be, they are avascular, meaning they do not have their own blood supply, but if they are well nourished, they do regenerate pretty easy, and you can see that kind of on the surface of your skin. And they are innervated, meaning that they are supplied by nerve fibers. So while they don't have a blood supply, you can feel every little paper cut you get. And if they're nourished, they're going to regenerate easily or reproduce rapidly. Each of your epithelia is going to have 2 names. The first name is going to tell you how many layers might be present, and the second is going to give you the shape of the cells. So as we head into talking about all the different types of epithelia, keep this in mind. Again, 2 names. The first is the number of layers, the second, the shape of the cell. Okay, so our simple epithelia in general focus on absorption, secretion, and filtration, because your simple epithelia are going to be a single layer, so very thin, and they're probably going to contain microvilli or cilia for increasing surface area, and they come in a variety of shapes. So first up, we have our simple squamous epithelium, and these are going to be a single flattened layer, kind of scale-like. You can see they look like they're just when you dump an egg, crack an egg into the pan to cook. So they just have this irregular shape. You can clearly see that nucleus. And here they're going to form membranes where rapid filtration occurs. So the air sacs of the lungs and the walls of the capillaries, you may find your simple squamous here. So, you know, kind of thinking about the lab test, thinking about this shape. It's an irregular shape. They're closely packed together, and you can see the nucleus. So there's not and you'll see as we move on, there's not a definite exact shape to this particular type of epithelia. Here we have both our simple cuboidal on the top and our simple columnar on the bottom. These are both single layers of cells. At the top right, you see a single layer of cube-shaped cells. These are going to be common in glands and their ducts. For example, the walls of the kidney tubules and the surface of the ovaries. So you're looking at one straight line of cube-shaped cells with the nuclei in all approximately the same position. And you notice the same kind of thing in the simple columnar, except they're tall. They're columns instead of cubes. And they're a single layer of tall cells fitting closely together. Again, the nucleus, that dark stained circular area, is all essentially in one line. This is going to be really important as you differentiate between some different types of column-shaped type cells. So with your simple columnar, you have that one even line of nuclei. So they're all kind of lined up in a row. And these are going to line the digestive tract and some of your cavities that open to the exterior. Now keep this simple columnar in mind as we move to the next slide. Remember I said that the nuclei are positioned in all the same relative lines. We've switched pictures and we have what looks like a little bit of a mess. And that's because we're now looking at pseudostratified columnar epithelia. This is cells that vary in length and they look like a mess because they overlap and their nuclei aren't in one single line. They're kind of all over the place. And this is relatively important because you get cells in a variety of lengths that are going to work for absorption and secretion. They're lining your respiratory tract. Well, that's kind of nice because you're talking about the ability to draw in oxygen, get rid of carbon dioxide, and the rapid transport that goes with those activities. So here you're looking at what kind of looks to be like more than one layer of cells, but really what's going on is that the cells are a variety of lengths and their nuclei are in a variety of positions. So you can easily recognize what's going on in the difference between a pseudostratified columnar and a simple columnar. And the pseudostratified columnar is a nice transition, even though it's not transitional epithelia, but it's a nice segue from our simple tissues, single layer tissues with nuclei all at the same kind of level, into our stratified epithelia. And here you're talking about 2 or more layers. And because they're 2 or more layers, they no longer absorb or secrete, so they focus on a protective role. They regenerate from the basal layers and push the cells toward the apical surface. We have a number of types of stratified epithelia as well. So we'll start with our stratified squamous, and this is the most common in the body. This is several layers, and they become flattened at the surface. So you'll notice in this picture they look quite obviously cube-like at the basement membrane, and as they move toward the surface, they become squamous. And that's because they work in areas of high friction like the esophagus, so those cells flatten and dehydrate, and then they slough off. You can have things like the skin where they're keratinized, so that's a protein that's going to strengthen that outer layer, or they could be non-keratinized. But here they start out cubes at the basement membrane. Cell division occurs at that basement membrane, and as each new cell is formed, it's going to push toward the surface, and the old cells are sloughed off, or they kind of shed off at the surface. And that's a nice thing because then as they slough off, they allow things that might create friction to slide by. The two pictures here are showing you an example of stratified cuboidal epithelia at your salivary gland at the top and transitional epithelium on the bottom. Your stratified cuboidal and stratified columnar are pretty rare in the body. They're found mainly in the ducts of large glands. With cuboidal, you get 2 layers with surface cells that are cuboidal, and your columnar, those cells are going to vary a little bit more in size and shape. Those are because they're less common, it is less likely to be tested on those too. And then we have our transitional epithelium, and this is a highly modified stratified squamous. And the reason for that is it's mainly in the urinary bladder, the ureters, and the urethra, so it's a stretchy type of tissue. So you have cells that can, when stretched, change shape. So think about that. That's good, because if you had rigid cells, you could absolutely only hold so much urine, and then you are essentially out of luck. A gland consists of 1 or more cells that make and secrete a particular product. The product is known as a secretion. So secretion is an active process, and glandular cells obtain needed substances from blood and transform them chemically, right? And so glands are classified according to 2 sets of traits: where they release their product and the number of cells. So they're really specialized secretory structures that produce and excrete whatever their product is that they need to have present. So we have endocrine glands, which secrete hormones into blood or tissue fluid, right? So they're ductless, and they diffuse directly into blood vessels. So this is the thyroid, the pituitary, some of those glands. Then we have our exocrine glands, and they have ducts, and they secrete out of cells to the body surface or to another cavity. So everything gets dumped through ducts to the epithelial surface. And here we're talking about things like oil glands, sweat glands, mammary glands, salivary glands, right? Things that are kind of have an exterior kind of obvious secretion. And we have kind of 3 means of exocrine secretion. Your merocrine is your most common, and they secrete by exocytosis. And we have apocrine glands, which aren't really found in humans. There's a nice little debate about whether or not they're human-based. And they kind of just accumulate below the free surface. And then our holocrine glands accumulate inside, and then they rupture. So sebaceous or oil glands would be an example of a holocrine gland. And they're kind of replaced by underlying cells, and essentially the cells here die for their cause, right? Holocrine secretion, so sebaceous. So those cells, like oil cells, rupture to the surface, and they die for the cause. So that's kind of what that's talking about. So this is just a nice, fun summary of most of the types of epithelial tissues that we talked about, right? This gives you kind of that animated view. I would spend some time looking at these different types and pictures. The photomicrographs are more like what you're going to see on the lab test. You won't see any of these pictures on the lecture test. There'll be questions about the different types of tissues, but you won't have to identify them by looking at them. That is a lab test 1 big deal. So spend some time looking at the pictures I included. Look at the photomicrographs in your book. Kind of come up with ways to identify one from the other. What are you seeing that is more kind of one tissue from the other? So we transition away from our epithelial and into our connective tissues. And connective tissues are really about doing exactly what they're called, connecting body parts. And we find connective tissues kind of everywhere. It's our most abundant tissue in the body. protection, support, and binding together of other body tissues. They have a structural and metabolic support piece to them. And they all develop from what's known as the mesenchyme, and that's your common embryonic tissue. All of your connective tissues consist of living cells surrounded by a type of matrix, and the differences between them are moving from rigid to soft, and there are kind of 5 main types, and we're going to go through those after we talk a little bit more about some special characteristics. So, your connective tissues have variations in blood supply. Most of them are highly vascularized, so they have a good blood supply. Some of them are avascularized, like tendons, ligaments, and cartilage, and that's kind of why they take forever to heal. And there's this increased extracellular matrix, and it's made up of 2 elements together that allow connective tissues to vary. So one component of the extracellular matrix is a structureless ground substance that's made of mostly water and some adhesion proteins, and then the other part is some kind of fiber. It could be a number of different fiber types. Collagen, so those are tensile, elastic, which are stretchy, and reticular, which are kind of fine for specialized organs. So the combination of the amount of these 2 elements, your ground substance and your fibers, is what's going to differentiate your different connective tissue types. So I'm going to work through these in a little bit different order than is in your packet. I apologize up front for that. I'm going to work through these from rigid to loose. So we start with our connective tissue types that have the most tensile fibers and the least amount of that waterless or structureless watery ground substance. And we start with our osseous tissue or our bone tissue. And this is, of course, our most rigid tissue. And obviously, if I said the word bone, we can kind of think about our bones. And they're made of all this osseous tissue. So our bones or osseous tissue are composed of bone cells in cavities known as lacunae, and they're surrounded by a hardened matrix of calcium salts and collagen fibers. They do have increased vascularity. The compact bone is the outer part, and the spongy bone is the porous, vascularized inner portion and leads to that central cavity. So if you're looking at the picture, the black dot, number 2, that's a canal down the center. We'll talk about it a little bit later. And then you can see all these concentric rings of spongy bone and compact bone as we work from inside to outside. And the cell here is known as an osteocyte. So our bone cells are osteocytes. So bone cells. Osteo meaning bone, sites for cells. This is our most rigid of our connective tissues. As we work our way from rigid to loose, we next come upon cartilage. This is a little bit less hard and more flexible than bone. Here we have what are known as chondrocytes. So they're cartilage cells. They're not as vascularized, right? So we mentioned on the kind of intro slide, we have a slow healing time here, but they do withstand tension and compression. And they do not have nerve fibers. This is why you can tear in cartilage and you may or may not feel that specific injury. We have 3 different types of cartilage here. I have included pictures. I'd spent some time for the lab test getting to know how to differentiate these 3 from one another. It is challenging, so spend a little time looking at it and figure out how you can differentiate them if they come up on the test. So we'll start with our hyaline. These are going to line the joints. They're kind of known as like articular cartilage. And you have things like the tip of your nose here, connecting your ribs to the breastbone, or from your ribs to your sternum. Your tracheal rings, the larynx, those are all hyaline. Fibrocartilage, we're going to start seeing more collagen fiber. They're going to resist tension and compression. Here you have the meniscus of the knee, your intervertebral discs, so those discs you find between your vertebrae. And then elastic cartilage is anywhere elasticity is needed, such as your external ear. So it's flexible but strong. You got an ear, you got an auditory canal. Those are types of elastic cartilage. So all three are cartilaginous. We're a little bit less tensile than bone, but we really don't have quite the flexibility to really do a lot with it yet. But if you like play with your ear right now, yep, go ahead, reach up, squeeze that ear. That is elastic cartilage. It has some bend, but in some places it doesn't feel as fabulous. So I've kind of been mentioning some of our connective tissue fibers, and I just want to make sure we understand your collagen is your tensile strength, so made of fibrous protein collagen. And then elastic is more of a rubber-like protein, elastin. And reticular, that specialized one, is really kind of a finer network of collagen for supporting. And as we move into some connective tissue proper here, we have both dense connective and loose connective. And dense has two different types. It has dense regular and dense irregular. I'm going to show you a picture next and talk a little bit more. But your dense connective tissues are collagen fibers with rows of fibroblasts. That's a specific cell type in that extracellular matrix. Dense regular are going to be tendons and ligaments. And your dense irregular is your skin. So the difference there is, while we're doing rope-like structures, your tendons and ligaments are strength along kind of parallel fibers, whereas your dermis provides strength in multiple directions. If you think about your skin, you wouldn't want it to just go one way. We want it to kind of do everything to kind of cover and support what's going on. So the lower layers of the skin are going to be your dense irregular, your tendons and your ligaments, parallel fibers, are going to be dense regular. Then your loose connective tissue is softer. We get more cells and less of our fibers. So we start to see a looser, kind of mix of things going on here. So you have areolar tissue, which is your most widely distributed connective tissue. It's going to cushion and protect your body organs. It's going to kind of wrap them up. We have our adipose tissue, which is fat, and it forms the subcutaneous tissue beneath the skin. It's going to be insulation and protection. It's also a food reserve and is stored in the cytoplasm. And then your reticular connective tissue is kind of more limited in where it's located. It forms stroma, which is going to support your free blood cells. It also can be found in lymph nodes and the liver and the bone marrow. So it's just kind of a soft internal skeleton framework that's there to support other cell types. So let's take a look at how we can differentiate between dense regular, dense irregular, and loose connective. So here we are, and I've left some description underneath each picture for you. And so remember I said your dense regular has got a parallel structure to it. You can now clearly see that. But it's still kind of wavy in appearance, which you're going to want to kind of take note of so that it isn't kind of getting confused with some of your muscle tissues that we'll look at in a second. So make note of what you're seeing here. Your dense irregular, you've got kind of a random orientation. It's going to kind of keep your skin from getting all stretched out, so to speak. And then you've got this loose connective, which has fewer clear fibers. So if you look at loose connective next to the dense regular, all that parallel arrangement is all collagen fibers. And when you look at the loose connective tissue, you have fewer collagen fibers. Those are the dark red stringy things that are stained in here, for a lack of better explanation. And you can start to see more ground substance, right? So you can see fibroblasts. Those are, again, cell nuclei that are stained black. But it's not a clear sheet at this point. It's kind of more spread out, and it may go in a variety of directions. I think loose connective is one of the easier ones to identify. because it's got kind of a distinct appearance in comparison to some of the others. And believe it or not, we round out our connective tissue types with blood. Yes, blood. Blood is known as vascular tissue. It's a connective tissue because it is cells surrounded by a non-living fluid matrix. We have cells surrounded by blood plasma. Very interesting. It's a fluid connective tissue, and it contains formed elements in a liquid plasma matrix. So fibers of blood are soluble protein molecules visible during clotting. Makes sense. And it is a transport vehicle for the cardiovascular system, carrying nutrients, waste, gases for respiration, etc. There are 3 types of cells. There are erythrocytes, which are your red blood cells. Lovely. Okay. Erythrocytes. And you'll get to study this, not with me, but in 202. So we have erythrocytes, red blood cells, and then you have leukocytes or lymphocytes, which are white blood cells, which can also be neutrophils. So you have 2 different types of white blood cells, lymphocytes and neutrophils. And then you also have thrombocytes. And that's pretty much the gist of that. So you have some different cell types, and you'll learn a lot more about blood when you do the cardiovascular system, or as a lead into the cardiovascular system when we talk about blood typing and how all that works. But just know it is your loosest, your fluid connective tissue, because you have cells surrounded by a fluid matrix. So it fits properties of the connective tissue. Our epithelial membranes are actually made up of both epithelial and connective tissues. They're continuous sheets of lining tissues, and there are 3 main types of membranes. We have our cutaneous membranes, which is referring to skin. These are keratinized stratified squamous epithelium for the epidermis, paired up with dense irregular connective tissue for the dermis. Okay, so you've got both an epithelial and a connective. You've got stratified squamous and dense irregular, and those are your cutaneous membranes. Your mucous membranes, known as mucosa, line body cavities that open to the exterior. There is no keratin here. They usually secrete mucus, and you're going to have stratified squamous or simple columnar with what's known as lamina propria, which is a loose connective tissue. So you've got stratified squamous or simple columnar epithelial with a loose connective tissue. And then your serous membranes are moist membranes of the closed body cavities. They secrete clear serous fluid for lubrication. You've got simple squamous with loose connective tissue. Examples of that are the pleura that line the thoracic cavity and lungs, the pericardium that lines the pericardial cavity in the heart, the peritoneum, which lines the abdominal pelvic cavities, and a lot of your internal organs. Okay, so cutaneous is stratified squamous with dense irregular. Mucosa is stratified squamous or simple columnar with lamina propria, loose connective tissue. And your serous is simple squamous with loose connective tissue. So on the next slide, I'm going to show you some pictures for comparison, but we're going to talk about our muscle tissues. These are our contracting tissues. They're highly vascular, and they're made up of 3 different types of cells. So we have our skeletal muscle tissue with our skeletal muscle cells. These are obviously attaching to the skeleton. They're under voluntary control. They form the flesh of the body. When they contract, they pull on the bones or the skin to create movement. The cells are long and cylindrical and multinucleate with obvious striations. Cardiac, you're going to find only in the heart. So they fit together at what are known as intercalated discs that form gap junctions. They're under involuntary control. The cells are striated, uninucleate, short, and branching. And then finally, we have our smooth or our visceral muscle, which is going to be found in the walls of hollow organs. And these organs are going to constrict or dilate with contraction. Some of them create like a wave-like motion, think like the digestive system. This is not striated, and it'll be more obvious when you look at the next picture. But they have a single nuclei, long, spindle-shaped, and also involuntary control. So here is a comparison, and your best bet, honestly, is to spend some time looking at as many things as you can to try to differentiate and recognize these. The photomicrographs in the book are good. The other PowerPoint's good as well. But you can see pretty clearly the difference. Your skeletal muscle are just sheets of striated or striped tissue, whereas when you look at cardiac, it's still striped, but it goes in a variety of directions. And then your smooth is not striated, but it is in pretty nice sheets. And so you just need to spend some time getting to know. Again, you want to look at the photomicrograph, which on this particular slide is on the right. And I think with your skeletal, it's pretty clean sheets of tissue. The way I would recognize cardiac is to realize that you're in striated or branching sheets, but you've got these intercalated discs that kind of horizontal line across the picture periodically. Those are your intercalated discs, and that kind of gives away this cardiac tissue. And then smooth muscle tissue, you just got to find ways that work for you to recognize and compare that to some of our other tissue types. Our final type of tissue is our nervous tissue, and we have two cell types. We have neurons. That's the top picture. That's a neuron. They're highly specialized, and they're there to conduct nerve impulses. And then the bottom picture is 6 different types of neuroglia, and we'll see this picture again when we get to the nervous system as you finish up the course. These are for supporting, binding, and insulating neurons. They're nourishment. They really kind of have limited division capabilities. Overall, nervous tissue is about irritability and conductivity. So the neurons with the supporting cells are going to make up our nervous system. And the neuron is pretty obvious. It's hard not to take a look at the picture that's in your book of the photomicrograph of the neuron and realize that that stands out significantly from the rest of the pictures. And last, but certainly not least for the evening, is dealing with tissue repair. And the body has a lot of ways it protects itself from uninvited guests or from injuries. We have mechanical barriers like your skin and the mucosa. You have things that line your respiratory tract. You have a chemical barrier in the stomach. And that means if you have an injury that these barriers have been kind of penetrated. And it's going to stimulate the body's inflammatory and immune responses, which are going to wage their battle in all these connective tissues. And you'll talk about inflammatory response much, much, much later. But tissue repair requires that cells divide and migrate. They have activities that are initiated by wound hormones. And we have 2 different ways that repair occurs. Either regeneration, so we replace destroyed tissue with the same kind of tissue, or fibrosis. And in fibrosis, we replace destroyed tissue with scar tissue, and scar tissue is dense connective tissue. One more time. 2 repair ways. Regeneration, where we're going to replace the destroyed tissue with the same kind of tissue, and fibrosis, which replaces destroyed tissue with scar tissue. Scar tissue is a type of dense connective tissue. Which of these 2 occur is going to depend on the type of tissue and the severity of the injury. Oftentimes you get both. So the process begins as inflammation sets everything up. We have the trauma causing inflammatory chemicals to be released, and the inflammatory chemicals are going to create leaky vessels, allowing white blood cells and fluid clotting proteins and plasma proteins to seep into the injured area. and the clot is going to seal off the injured area and prevent anything from getting in, and the surface of the clot exposed to the air dries and forms a scab. That's why you don't want to pick the scab. Scab becomes your barrier to bad things getting inside. So that's the first picture, right? We get inflammation, we get leaky vessels, we get clotting, and we get a scab. As we move forward, things are going to get organized to restore blood supply, clot gets replaced, and the fibroblasts are going to multiply, produce growth factors, and the fibroblasts will then revert to kind of a resting stage, or undergo what's called apoptosis, which is essentially cell suicide. So the regenerating epithelia, you can see it starting to rebuild from deep to surface in that middle picture. And then in this particular case, you get regeneration and fibrosis. This is a cross-section of a non-extensive skin wound, and it encompasses both regeneration and fibrosis. So the fibrous area matures and contracts, pulling the margins of the wound together. And as it regenerates, the epithelium thickens under the scab, and the scab will detach. And a fully regenerated epithelium with an underlying area of scar tissue is the result. Scar tissue may look like that white line you see in that picture, and it kind of depends on the severity of the wound. All right, so 3 pictures. Step 1, inflammation, leaky vessels, clots, and scabs. Picture in the middle, we begin some healing process from the deep end, and we get rid of debris, and apoptosis occurs and we get some cells that go away and they don't need to be there, and get regeneration of whatever destroyed capillary you got going on, etc. And then in that final picture, you get regeneration and fibrosis. And again, the scar tissue or the white line kind of depends on the severity of the injury. So as we finish off chapter 4 and you have everything you need for lecture test 1, we just want to close up by making sure that with this PowerPoint you understand that a lot of this is super supportive for lab. Pictures and identification will be on the lab test. That's not what gets covered on the lecture test, but I'll give you more information right before the test. And the big ideas here in chapter 4, we're understanding the different tissue types, that we've got 4 tissues, and most of them are found in organs and together. So we have nervous, muscle, connective, and epithelial tissues, making sure that you understand what makes each one different from the other and unique. And then we closed with tissue repair. Make sure, I probably will open a Q&A board eventually, if I haven't already, based on when you're watching this, for questions as we lead into lecture exam 1 and then lab test 1. And I am always available if you would like to email me or use the PickTime app web piece to book a meeting with me if you would like to have a chance to have a meeting. So with that, that's the end of first lecture section, lecture part 1, chapters 1 through 4, ready for lecture test number 1.